Dear students, welcome this class of genetics. Today we'll start paper of molecular biology and begin with we will see genetic code and its character. After completing this lecture, we should be able to know what is a triplet code, what is a comma-less code, what is a non-overlapping code, what is meant by the degeneracy of a code, and what are start codons and stop codons, and what is Wobble hypothesis. So first let's begin with the function of the genes, which is defined as the unit of DNA. What is the function of a gene, which is called as the unit of a DNA, is to synthesize the proteins. Evidence for the function of the genes came from studying the fungus Neurospora crassa by George Beadle and Edward Totham. The famous Beadle and Totham experiment in 1941, which gave us the one gene, one polypeptide hypothesis. Today it is called as one gene, one polypeptide because one gene is responsible for synthesis of one protein. Beadle and Totham, they proposed for the enzyme arginine pathway, that it had a separate gene for its synthesis. They proposed one gene, one enzyme, but later on this enzyme was replaced by polypeptide. Central dogma of protein synthesis, which we all know in biology, is the flow of information which takes place for creation of an individual organism. The information flows in the form of DNA, in the DNA in the form of the arrangement of nucleotides, gets converted into RNA, which in turn gets converted into protein. So the first step of conversion of DNA into RNA is called as transcription, and the conversion of the information from the RNA to proteins is called as translation. So this is the central dogma of protein synthesis, and it is well visualized here by this flow sheet. So I'd like you to just concentrate and make this picture permanent in your brain. From here onwards, if you want to study genetics, or if you are interested in any type of mechanisms going out in genetics, you should know this as the core function of biology of any genetic systems. This is the center stage. This is the headquarter of cell. This is the process which is going out continuously, which creates an individual organism with its particular characters. So as you can see, DNA in green here, a DNA molecule, you are well informed about its structure, so I will not go into much detail. You know they contain nucleotides, adenine, thymine, guanine and cytosine. When it separates, it gets copied into a mRNA. The process is called as transcription. You can see the blue ribbon here. This is mRNA formation. The process is transcription of DNA, where encoding of or copying down of the nucleotides present on the DNA into mRNA takes place. The exception is there that there is, instead of thymine, there is uracil molecule. So adenine, uracil, guanine and cytosine. So this blue ribbon, it is a single strand molecule. In the cell there is an amino acid pool, okay, as good as there is a nucleotide pool. The nucleotide pool is always filled with cytosine, guanine, adenine, thymine and uracil. It is continuously giving these nucleotide supply to the newly formed genetic materials like DNA, mRNA, rRNA, tRNA, etc., whichever may be needed. So this transcript, which is called mRNA, assembles the amino acids present from the amino acid pool, which are 20 in number, depending upon the nucleotide arrangement present on the mRNA. So this transcript, or the mRNA and the nitrogen bases present on it, will be the focus of study in today's lecture. That arrangement is called as genetic code. Whether that has to be read in triplets, or whether that has to be read in quadruplets or singlets or doublets, we will see. Of course, you know it is a triplet code, but how this came into existence, it has a very interesting history. Those who are interested can go dwell into much more detail and see the fascinating history, how it was deciphered. So unfortunately that detail is not there in your syllabus today, but I would like to explain the characteristic features of this nucleotide arrangement on the mRNA. So the central dogma is transcription, formation of mRNA. Translation is assembling these amino acids into long chain mediated by an intermediate adapter molecule called as tRNA. So this is these three anticodon containing structures bringing one amino acid each are called as tRNA. So as many as amino acids are there, that many tRNAs will be present in the cytoplasm. Maybe double amount. So 20 amino acids are there, 40 tRNAs will be there, which have sustained in the evolution. So this process is translation, and the polypeptide synthesis is the protein synthesis. So this is the central dogma: transcription, translation, and when a DNA doubles itself, replicates itself, it forms DNA. So today we are talking about this blue ribbon and its arrangement of nitrogen bases, which are called as codons. That is the genetic code. So how was this code deciphered? How was this genetic code found? Deciphering the genetic code required determining how 4 nucleotides, that is A, T, G, C, could encode more than 20 amino acids. It was firstly observed by Francis Crick and Sidney Brenner, who determined that the DNA is read in the sets of 3 nucleotides each for 1 amino acid. Later on, Marshall Nirenberg was the scientist who deciphered the genetic code in 1961, and a scientist of Indian origin, Hargobind Khurana, created new methods to produce synthetic nucleic acids. Again, that is the characteristic features of the genetic code. Some of the common properties of the genetic code, if you see, this genetic code is made up of triplets. It is read in triplets, and it has unique features. It has universality. We will see that characters. Secondly, the genetic code is a non-overlapping code. Genetic code is comma-free code. Genetic code is degenerate code. The genetic code has a polarity which is read by the tRNA, and the amino acids are bought, assembled to form polypeptide. Genetic code contains start codons and stop codons, which start codon which codes for a specific amino acid, and stop codons are nonsense codons which do not code for any amino acids, which terminate the protein synthesis. This genetic code are universal in nature, means from a housefly to a Escherichia coli to a guinea pig to you, these 20 sets of these 20 amino acids code for the same codons universally. Just to give you a fair idea of what a reading frame is and what makes it a sensible strand and what are the codons, why they are called as nonsense codons. Later on, these terminologies will be very frequent for you in the coming days. So just to give you a fair idea, you see, she can fix the hat. This is a sentence made up of, each word is made up of 3 letters. So 3 in a particular order is making some sense. Now if I just add n after n and f of can and fix, and the rule is that it will always be read in 3s from the starting point. So SHE3, CAN3, then as n is added here, NFI, XTH. So suddenly the sensibility of a reading frame is lost due to addition. This is what happens in mutation. That is not the point of discussion here, but just think about it. Then add one more, 3 additions, then suddenly it gains some meaning, though the meaning has changed. The sense is there, but the meaning has changed. Sensibility is achieved. This is reversing the mutation, but the rule is that in 3s. So therefore it is called as a reading frame. So the same thing happens with deletion, the same thing happens with addition, and this is how the sequences on a mRNA make some sense. Here the sense is understanding of the word, there the sense is in a triplet, 1 amino acid is bought. So codon can be defined, or a genetic code can be defined as a set of 3 nucleotides which specifies a particular amino acid on from a mRNA, when coding from a mRNA. The requirements for this are tRNA and rRNA. These things we will study in great detail in our next unit. But here just see the characteristics of the features on a mRNA. The nucleotide arrangement on the mRNA is called as the reading frame, and in triplets this will be called codons. So reading frame is the series of nucleotides which is read in the sets of 3 codons. Only one reading frame is correct for encoding the correct sequence of amino acids. Means, for example, if I am reading this sentence, reading frame the series of, so this is the frame which starts with R, but suddenly if I start with ADING, adding frame, so on, then that frame can also be correct, but for that set of frame only. All this is determined by which is the first codon which is read. That is called as the initiation codon or the starting codon. Again, there is universality, that all organisms, all the organisms in their DNA, they start with a reading frame of AUG. All the life systems, they start with AUG. So AUG is called as a initiating codon or the start codon in code. So how this code is a triplet code? This is one of the first characteristic features of the the nucleotide basis on the mRNA, that the triplet code is responsible for one amino acid, means the genetic code is a triplet code. See the possibilities here, a singlet, doublet, triplet and some quadruplet mathematics I will try to show you. If the code was, hypothetically, why we are believing that, or it was deciphered with scientific experiments, but before actual experiments of Nirenberg and Matthew, Hargobind Kuranberg, then Hargobind Kurana and many other scientists, they first did the work on paper. It is simple mathematics. See, if the codon was a singlet code, then one nitrogen base would have code for one amino acid and there will be only 4 codons. A would be one codon, G, U, C, T, or 4 or 5 codons at the most. Now, there are 20 amino acids. How will these 4 codons compensate with 20 amino acids? Only 4 will be bought. These are 4 codons and one brings one amino acid, and it is specific. If A brings leucine, it will not bring any other amino acid throughout its life. So it will bring only 4 amino acids from the pool, or it will be responsible for accumulating only 4 amino acids from the pool. What about the remaining 16? When you take a protein and see the protein change, you will find 20 different amino acids in a polypeptide chain, any polypeptide chain. So maybe then it is a doublet code. That how, what is a doublet code? You see A, G, C, U on this first base and second base A, C, G, U again. So multiply, A will be 1, AC will be 1, cross multiplication tabulation results into 4 doublets, 4 raised to 2, 4 into 4, 16 codons. Maybe, now this may be nearer to reality, that 20 amino acids and 16 codons. But 16 codons, 4 amino acids are still left. These 4 amino acids are found in protein chain. Means again, this is not the possible combination. So let us go for a triplet code. Now triplet code is 4 raised to 3. 4 are the nitrogen bases when it has a permutation combination of 3 levels. That is the first base, second base and the third base. Then you have, you see, this is the red is the first base, blue is the second base and green strip is the third base. In green you have 4 combinations. So U will be 1. Here this is 1. UUC will be second. UUA will be third. So on and so forth. Each box will result into 1, 2, 3, 4 codons. Like that you have 1, 2, 3, 4 boxes. So it 4 into 4, 16 into 4. 64 codons will be there. 4 into 4 into 4, 64 codons will be there. Now again this is a dilemma, 20 amino acids and 64 codons. So what about the other? Now the amino acids are being taken care of. Number has overpowered, the codon number has overpowered the amino acid number. Now see here, if you think about 20 codons coding for 20 amino acids, still you have 44 codons which are wasted. Okay, anything above this, if it is a quadruplet code, then the number of code will be, you see, singlet 4, doublet 16 codons, triplet 64 codons, quadruplet 256 codons, pentaplet 1024 codons. So above triplet, quadruplet and pentaplet codons are out of question debatable issues, because non-debatable issues. Why? We are already having 64, which you have to think now. 16 and 4 codons is out of question, because even in 16 there are 4 amino acids which are found in the protein chain. So this is accepted hypothetically. Let us hypothetically consider that the codon is a triplet code which converts the number of codons on a mRNA chain into 64 possible type of permutation combinations. Means what, कितना भी combination अगर आपने लगाया तो 64 हो जाएंगे triplet. Above that there is no possibility, because the number of codons goes on increasing. So it was believed that it is a triplet code. And what happens with that 44 codons? You can see here, there are certain amino acids. Let us take case of proline, serine, threonine, alanine, the second column. You see the whole second column here, and up to a great extent the last column also, glycine, arginine. Okay, what is unique of this? Here see the second column, first row, UCU codes for serine, UCC codes for serine, UCA codes for serine. Means there are more than one type of codons which code for the same amino acid. This is called as a redundancy of a code. This is called as the code is redundant or degenerate. I'll come to that. There are more than one types of codon coding for the same amino acid, and this is explained by the Wobble hypothesis. If you can understand this, you will understand Wobble hypothesis. So first here you just understand that the code is a triplet code. This triplet code or the 3-letter code was first proposed by physicist Gamow, but he had a different concept, and that was called as the diamond code concept in 1954. Nevertheless, he gave some features which are well accepted today by physicist Gamow. The codon of the present day genetic code specifies amino acid in a polypeptide chain is made up of sequence of 3 nitrogen bases on the mRNA in a specific sequence, as you can see here in the figure. This mRNA chain is clubbed into 3 codons, 3 nitrogen bases, and 3 nitrogen bases forms 1 codon. The first experimental evidence supporting the concept of triplet code was provided by Watson and Crick himself and his co-workers in the T4 bacteriophage. And now today it is a worldwide accepted fact that the mRNA harbors nitrogen base arrangements in a triplet code fashion. So as you can see here, this picture or this table is universal, and there are 4 codons. The first letter, this is the second letter, and this is the third letter on the mRNA. So you see the color, blue color, UU, and the blue color, this will phenylalanine, UU and C, UU and A, so on and so forth. You have 64 codons. Each triplet nucleotide sequence of the codon here refers to the nucleotide sequence in the mRNA. Remember that, not DNA. You should always remember that codons are the nitrogen base sequences present on the mRNA, not the DNA. And this specifies the incorporation of amino acid or a polypeptide chain. And there are 2 types of codons. Here you can see AUG is a methionine coding initiator codon, and there are 3 codons, UAA, UAG and UGA. These are the stop codons or the nonsense codons which do not code for any amino acid. And remember this, we will see in protein synthesis, when the ribosome moves, when there is no addition of any amino acid, the whole protein synthesis terminates. So this is the universal table. If you watch it, you will get a fair idea of how many amino acids and how many codons are there. So this is how the functioning of codon works. The triplet codes on the DNA. Strand of the DNA or a part of a gene is converted into a mRNA by transcription, and triplet code CCA codes for first amino acid, TCG codes for amino acid 2, depends upon where the first amino acid is incorporated by the initiating codon. Note here the precise order of the amino acids, they are joined. Eventually this is translation, and the amino acids are joined to form a protein polymer, which forms the part of protein synthesis discussion later on. The second characteristic feature of the code is that it is a comma-less code. A comma-less code means that no nucleotide or commas are present in between the 2 codons. As you can see here, AUG is 1 codon. there is no comma. Immediately the next successive is a CAG, so on and so forth. Code is a continuously read without any overlapping or without any comma. There is no nucleotide which is left as a comma, or there is no punctuation mark like of any nucleotide extra which will be left and then read. It is read in a continuous fashion. Code is continuous, therefore commaless, and no letter is wasted between two words or two codons. So as you can see here, this 1 2 3, then there is hypothetically think this amino acid as a comma, then 1 2 3 comma 1 2. No, this doesn't work like this. It works like 1 2 3, then the fourth will be 1 2 3, then the seventh will be 1 2 3, 10th will be 1 2 3, like a continuous reading without any hindrances, without any full stops or commas. It is read. Second, it is a non-overlapping code. I will come to that. But the code is commaless and non-overlapping. You should say in the same breath, because this is even if I am telling 2 points here, that is one and the same point. The code is a non-overlapping code. You see, if it was a overlapping code, what would have happened here? CATGATC. Okay, CATGATC is a sequence. Let us just consider. If the code was a overlapping code, CAT would have formed one amino acid, then ATG would have formed another amino acid, then TGA would have formed another amino acid, then GAT would have formed another amino acid, ATC would have formed another amino acid. So the same letter is not used again in the formation of more than one codon. Once CAT is used, finished. No codons, no nucleotides from this codon is used. The next, immediately the next without any comma will be used, and that forms the second codon. So the code is a non-overlapping one. Remember this thing. This doesn't mean that ATG will not be a codon. If ATG is the first codon, the second codon will be ATC. C will be left there. Okay. So how this statement gains significance? It doesn't have commas. It doesn't have commas. It is non-overlapping in its reading function. See here, as I said again, the codon is a non-overlapping one. AUU forms amino acid 1, GCU forms amino acid 2, CAG 3, so on and so forth. If it was overlapping one, AUU would have formed one amino acid, UUG would have formed another amino acid, then UGC would have formed another amino acid. Even if the third letter is not completed, you would have had 3 amino acids in the polypropyl. But it doesn't function that way. It is a non-overlapping code and gives rise to addition of amino acids with amino and carboxyl end, which are very perfectly insequentially joined. This will be detailed in the protein synthesis. So genetic code, remember, is non-overlapping. Once the reading is commenced, it has started at a specific codon, which is AUG. Remember, it always starts at AUG, which is the starting codon. As it starts, there is no punctuation marks between the codons, and the message is read in a continuous sequence of the nucleotide triplets until a nonsense codon is reached. This nonsense codon doesn't add any amino acid, and termination of protein synthesis takes place. The fourth important point is the code is degenerate one. A given amino acid may be coded for more than one codon. As I mentioned, there are 64 codons and only 20 amino acids. So some amino acids are coded for by more than one type of codons. So you can see here, valine, it codes for 4 amino acids, lysine codes for valine is amino acid which codes for 4 codons, lysine is amino acid which codes for 2 codons, tyrosine is amino acid which codes for 2 codons. So the occurrence of more than one codon for a single amino acid is called as degenerate or degeneracy of a code. If you see the genetic code dictionary, means all the arrangement of nucleotides on the mRNA, we will see that most of the amino acids, they have more than one codon. Out of 61 functional codons, 3 are nonsense codons. Out of 64, 3 are nonsense. 61 functional codons, AUG and UGG are the only two codons, AUG and UGG, which code for only one amino acid. Remaining all, they have more than one, what you can say, remaining amino acids, they have more than one codons. They are coded by 59 to 60 codons. Okay, so that is degeneracy of the code. As you can see here, the amino acid are mentioned, the same table is there, but you can, to be more clear in the picture, amino acids are shown. So UUU, UCC, and these are these codes for phenylalanine, serine, proline. Color coding is there, you can see the color coding. And leucine actually codes for 6 codons. One leucine molecule codes for 6 codons, you can see that. Okay, so you go through this table, you will get a fair idea of what degeneracy of the code is. Now there are start codons and stop codons. Stop codons are the codons which do not code for any amino acids, and they are helpful in terminating the protein synthesis. These are also called as nonsense codons. So 3 codons, UUA, UGA and UAG in the genetic code, they are used to terminate the translation process, and therefore they are called as nonsense codon. Similarly, codon AUG is used to signify the start of a translation process, start of assembling of amino acids on the ribosomes. So this AUG codes for amino acid methionine, but when it is starting molecule, N-formyl methionine is the amino acid. We'll see that in our future discussions. The remainder of the code is degenerate, as I already mentioned that more amino acids, that some amino acids are specified by more than one type of code. Now finally, the most important part of today's discussion, that is the Wobble hypothesis. Wobbling is an English word which means to remain unsteady or to move away. As you can see, this is a mRNA chain. See the picture over here. This is a mRNA chain, and the tRNA and the blue color is amino acid. So one tRNA which contains anti-codon site. So if there is, let us say, TTT codon, A codon, there is no T on the RNA or the mRNA. There is, let for example take AAA, adenine. This tRNA will come with UUU, and it will bring a amino acid specific for AAA. There will be UUU and amino acid. Now the third base which is there, this does not follow the Watson and Crick pairing. What is the Watson and Crick rule? Adenine pairs with uracil, guanine pairs with cytosine in protein synthesis. Okay, generally is A with T, G with C is DNA, and A with U and G with C in RNA in the protein synthesis. So this third base, there are more than one codon for one amino acid. We have seen this in the degeneracy. Now to explain the degeneracy, then how are this managed? How the amino acids are 20, tRNAs are 40, codons are 64. How does this match? To explain this possible case of degeneracy of the code in 1966, Francis Crick, he proposed the Wobble hypothesis. According to this hypothesis, only the first 2 bases of the codon, they follow a precise Watson and Crick pairing. That is, only the first 2 bases of the codon with the anticodon of the tRNA have a correct Watson and Crick pairing, while the pairing between the third codon may wobble. This is the wobble, unstable, not stable. This phenomenon permits a single tRNA to recognize more than one type of codon. Think about it. We have seen just now one amino acid which one amino acid coding for 6 codons. So if there is, for the 6 codons, there is only one tRNA. Now see, interestingly, I will go back to this example of leucine here. You see leucine here. What is the characteristic feature of leucine? See, CUU, CUC, CUA, CUG. First 2 bases are common for all. The third base is different. So if there is one tRNA which codes CU, there is GA, and whatever may be the third, GA, TC, whatever is the third one, it is wobble. and it is bringing leucine. So one tRNA which will not consider the third base, only one tRNA can bring each time leucine for all these 4 codon, isn't it? So that is degeneracy of a code is the possible explanation for degeneracy of a code is Wobble hypothesis by the tRNA. So this Wobble hypothesis phenomenon, which was discovered by Francis Crick in 1966, remember, most of them they confuse with telling, I have been into this teaching business for more than 20 years and I hear the nonsense that Wobble is a scientist. I don't know how this myth comes into students' mind. Those who don't study, they say Wobble hypothesis was proposed by Wobble. That is not something what you can say, that is very funny. Wobble is a simple English word which means to sway away, to unsteadiness, or which is not stable. Okay, when this wobbling permits a single tRNA to recognize more than one codon, therefore, as you can see in this picture, although there are 61 codons for amino acids, the number of tRNA is less than that. There are around only 40 tRNAs, out of which active still less are there, and this can be carried out by wobbling. The Wobble hypothesis, it states that in this example, as you can see, a leucine molecule is brought, CUC, GAG is there, GAG. Now GAG, the third pair, first is C, second is U, third is C, first is C, U, Q. Now GU is not a normal pairing, but temporarily it just continues with that. It is a wobble pairing, but the amino acid for CUU, which is what is not correct, it is correct. So the Wobble hypothesis, it states that the first 2 bases of the codon, they make a normal Watson and Crick pairing. A temporary hydrogen bonding is there. tRNA structures are not permanent structures. They just come, sit there to release the specific amino acid for this code. Okay, and then they move away. They again bring from the nucleotide pool, they bring one amino acid, put it here. So as long as this third base may be correct or not, only the amino acid has to be correct for this codon. There is no issue in the whole mechanism of metabolism. Okay, so the first 2 bases are normal Watson and Crick. This is also temporary, the third is also temporary. The remaining position, the third position, is not strictly Watson and Crick pairing, it is called as the Wobble pairing. Wobble hypothesis thus proposes a more flexible set of base pairing rules at the third position of the codon. Okay, so this is the Wobble position. The Wobble base pair is a pairing between 2 nucleotides in RNA molecule which does not follow the Watson and Crick pairing. There are 4 types of possible Wobble base pairs, which are those: guanine and uracil, GU, hypoxanthine and uracil. Hypoxanthine is a molecule, is a nucleobase of inosine. Inosine based nucleotide it forms. It is also one present in the cell, and hypoxanthine and uracil, they are also formed. There are tRNAs which contain inosine molecule at their third base, that is hypoxanthine, and it recognizes uracil as well as adenine as well as cytosine. So hypoxanthine molecule can recognize uracil, adenine and cytosine as a Wobble base pair. This is present in the tRNAs. Okay, so inosine displays the true qualities of Wobble in that if it is the first nucleotide in the anticodon of the tRNA, that is the first one, then any of the 3 bases in the original codon can be matched with the tRNA. So in the wobbling of the tRNA, inosine plays a true quality of Wobble hypothesis, and it is the one which carries out the protein synthesis. That is, inosine containing tRNAs are very efficient in recognizing the Wobble position. So how does this Wobble has some significance at the molecular level? Evolution has made this possible. First of all, remember, whatever the genetic mechanisms we are studying or we will be studying in this semester and the next semester, you have to understand one thing: all this which is natural is generated from evolution. Evolutionary mechanisms have resulted into these mechanisms, these processes, and that has evolved into a character or a trait. Those processes have made us evolve into what we are today. So Wobble base pairs have shown to facilitate many biological functions, most clearly proven in the bacterium E. coli. It is well understood in E. coli. The thermodynamic stability. Any reaction which goes on should have a thermodynamic stability. Then and only that reaction will continue further in a decided way. And this thermodynamic stability of a Wobble base is as good as the Watson and Crick pair. First thing, Wobble base pairs are fundamentally very critical for the translation process, and they provide a very stable RNA structure. Yes, RNA secondary structure. As I said, the inosine molecule is there. So this stability of, when will a molecule be stable? When it carries out the function efficiently. If it is inefficient in its function inside a cell, evolution will just wipe it out. Remember this thing. So Wobble base pairs are fundamental in RNA secondary structures and are critical for the proper translation of the genetic code. Wobbling also allows the faster dissociation of tRNA from mRNA and also protein synthesis. Association and dissociation are very important processes in the formation of a protein. When it is ended, it should dissociate and should not undergo wasteful synthesis. A cell never undergoes a wasteful synthesis, and the more faster the dissociation of the tRNA from the mRNA, that is tRNA, this is called as a uncharged tRNA. Once it leaves the amino acid, it is uncharged, it should leave, it should dissociate, go to the nucleotide pool, bring another amino acid and be ready for the next step. So this association and dissociation of the tRNA for continuous elongation of the polypeptide chain, wobbling allows it faster. If it was a permanent, then imagine the thermodynamic reaction would have slowed down. It would have been somewhat slower if 3 bonds were formed at that site. Only 2, no mismatching, process is faster. Last point, the existence of wobble minimizes the damage that can be caused by misreading of a it does not allow mutations. For example, the leucine code CUU, if it is misread CUC or CUA or CUG, if there is misreading during transcription, then the codon would still be recognized for leucine amino acid because the second base is wobble base. So this is the significance of wobbling, and this is all about Wobble hypothesis. This is all about the characteristic feature of the genetic code. In our next lecture, we will see the structure of RNA polymerase and the process of transcription. While seeing this lecture, I have an important note for you. You can use the pause button as you wish. When you want to read the slide, pause the lecture, pause the video, read the slides. While reading, don't listen to me. While listening, don't read the slide. This is an important trick to understand, and simultaneously pause it and take down the notes which you understand. This is the way to listen to a lecture. If you are not with a pen and paper, then my dear friend, you have not got anything. Always listen to anything, anybody with a pen and a paper. Take down the notes. Pause wherever necessary. Take down the notes. Listen when you are not writing. Write when you are not listening. Do one thing at a time. Do it properly. Understand genetics is fun. Genetics is interesting, as we will see in our further. That's all. Thank you.